Hello, back for chapter 8, part 2. So now let's talk about some changes in genetic material. Let's talk about what's a mutation. It's a permanent change in the base sequence of DNA. Okay, so that can happen due to mutagens, which are substances that can cause mutation. Some drugs can cause mutation. Radiation can cause mutation. So there are different things that could lead to it. Or they can occur spontaneously when they don't need a mutagen to be present. Mutations can be neutral, beneficial, or harmful. And I found this very easy to follow diagram that talks about all the types of mutations. So let's look at them here. So this is the DNA level, mRNA level, and protein level, right? DNA, RNA, and then protein. So this is the guy with no mutation. So the DNA of this organism has a lysine. What is a lysine? It's an amino acid that is coded by AAG in the mRNA level and TTC in the DNA level. Does it make sense? T pairs with A, C pairs with G. Okay. What would be a silence mutation in this case? Remember that the genetic code is redundant. There are some bases that are repeated. It's called generated. So there are some bases that are repeated in the 3-codon system, or not repeated, that are changed, that don't change the amino acid. For example, TTC codes for lysine, but also TTT codes for lysine. So if I change this C by a T, and then I have an A in mRNA level, I still have lysine in the end. Got it? Okay. So what would be a nonsense mutation? So these are all point mutations, right? They happen to only one base. You could have mutations that happen to more than one base. A nonsense mutation would be changing this T, this thymine, by an adenine. And in that case, the pair would be uracil, right? Well, this is a stop codon. What's a stop codon? It's a codon that when that codon gets in the ribosome and brings a transfer RNA to it, that codon, UAG, makes the translation stop. Okay? It doesn't continue. It's usually present in the end of the sequence for that protein. So then it stops. Okay. What would be a missense mutation? Instead of the T, you would get a cytosine, and then you would get a guanine in the mRNA level. And that AGG codes for arginine. Is arginine a different amino acid? Yes. So you have a mutation that changes the amino acid. Is that going to code for that same? So all these genes that suffered this mutation, and now instead of lysine in a specific position, you have an arginine. Is that going to be the same protein in the end? No. But the fact that you're still replacing it for a basic, you see how purple are basic, for a basic amino acid, could not be, excuse me, it could not be a very deleterious, prejudicial type of mutation. But if instead of your T, you replace it for a G and not for a C, that is the guanine, then you would have a C in the mRNA level, and ACG is the code for threonine. And threonine is acidic. And because it's acidic, what's happening? You would have a much bigger difference in the structure of that protein because you're changing for a very different amino acid that has a different type of charge compared to the one that was originally there. Okay? Does it make sense? There is a lot more to this subject. I just want you to know the basics, okay? So mutations can happen if drug-induced or just spontaneously. your book has an image that just shows exactly what I told you, right? Something is replaced by another one, changes the amino acid. If it's a silence mutation, it's not going to change the amino acid, and so on. Now we have two different types of gene transfer that are not mutations. They're called genetic transfer. So genetic recombination is the fact that you exchange genes between two molecules of DNA, and that creates diversity. Do we as humans do this? We totally do this, right? How do we do this? By crossing over. What's crossing over? Two chromosomes break and rejoin, resulting in the insertion of foreign DNA into the chromosome. Look at that. You have a lavender and a pink DNA, right? Let's say purple and whatever, purple and pink. A piece of the purple is cut through an enzyme that is called recombinase A, and that enzyme cuts a piece of the pink one, and exchanges. One piece of the pink goes to the purple, one piece of the purple goes to the pink chromosome or gene in that chromosome. Are you going to have a lot of different translation here because you're exchanging it? Yes. And if this cell is dividing, instead of having all purples and all pinks, you're going to have a cell that has all purples with a piece of pink, and another cell that has all pink with a piece of purple. So if here there was a gene for, like, I don't know, hair color, this daughter cell is going to have a piece that is coding for dark color, and this one is going to have a piece that codes for light color. I'm just being very simplistic here, but just so you understand that it would increase the variability in how these genes are expressed, okay? Or depending on the type of relationship that this gene has with its allele, which are, where's my pen? Hold on. My pen is not wanting to work right now. Okay, so for example, there is an allele for this gene here, right? So if these two genes combined would give a certain trait A, let's say whatever A is, and the combination of these two would give a different type of trait, right? So you would have more variability here. So that's the purpose of crossing over. So crossing over and genetic recombination can happen within the same cell, but you can still have recombination by horizontal transfer. Remember how I told you about the 3 mama cells here? So let's make them again. 1 bacterium, 2 bacterium, 3 bacterium. They're all having babies. 2 babies here, 2 babies here, 2 babies here, right? What's going to happen to these babies? That's the vertical transfer. They're going to get the genetic material from their mom. But remember that the genetic material from the mom could also, so let's say these are all different mommies, right? These are 3 different bacteria. They could be the same species. They could be a different species. And this one is a different type of DNA here. So what can happen is that this cell can give a little piece of genetic material to this cell, or vice versa. This cell could transfer genetic material to this neighbor cell, and vice versa. So do you agree that what we're talking about now is horizontal gene transfer? I hope you do, because it's true. So horizontal gene transfer can happen through 3 different processes that are called transformation, transduction, and conjugation. So that's what we're going to talk about now. Transformation is when you transfer genetic material from a bacterium to another, but the bacterium that donated that piece of genetic material is not alive anymore. And that genetic material just fell in the environment, and that's what we call naked DNA. It's DNA that was like from a cell that just died, and another bacterium can incorporate it. Okay? So this is an experiment done by a scientist called Griffith, and he showed something super interesting to prove transformation. So this is what he did. He had, I need my pen again. I need another color. Let's try yellow. He had these bacterial cells with capsule. Okay? So that has a capsule. These bacterial cells with capsule, so remember, capsule confers virulence, because the macrophages cannot, or it's much harder for a macrophage to phagocyte a bacterium that has capsule. It acts like a wet soap. So the phagocyte cannot get the bacteria. The macrophage cannot eat it. So then you don't neutralize the bacterium that infected your body. Where is my pen again? Here we go. So when you inject encapsulated bacteria, mouse died. And then you isolate colonies of this bacteria from this animal, and you see that they were all encapsulated. You inject non-encapsulated bacteria, so less virulent, into this animal. Animal is still healthy. A few colonies of non-encapsulated bacteria were isolated. Phagocytes could destroy this bacteria because they don't have capsule. If you hit kill encapsulated bacterium, they don't do anything and the animal is still healthy. If you get these encapsulated bacteria that were hit killed and you mix them with non-encapsulated bacteria that are alive, animal dies. What does that mean? Can you think about it? So, I'm going to get another color here. It means that the gene from the encapsulated bacterium, the genome, right, has a gene that codes for making capsule. This says capsule, right? And that gene was incorporated as naked DNA because this cell was dead, right? So this DNA was in solution here, and it was captured by the non-encapsulated bacteria that became capsulated because it could make that gene, that protein that would make the capsule. You may say, but capsule is sugar. You're talking about protein. It would make the proteins that are enzymes that catalyze the formation of capsule, which is sugar. Got it? Okay. And then, because capsule is virulent, it gives virulence to that pathogen, the animal dies. So what is the process that we're learning here? It's this process. Transformation. So transformation is when you have DNA in solution in the environment and your recipient cell that doesn't have that sequence that, for example, codes for capsule, gets incorporated into that chromosome, and now this bacteria has the property conferred by that gene that was in the environment. Do you agree that this is horizontal? It doesn't come from a mommy to the daughters. It came from the environment, so horizontal transfer. Conjugation. Conjugation is like a kiss. Look at that. This is what we learned. Remember when we learned in chapter 4 that this is what we call pilus or pili? So a sex pilus or pili, plural, because bacteria can have more than one, makes a bridge. And there's also another type of structure that is called mating bridge, which is shorter in length, that allows for transfer of the genetic material from a positive cell, you see how it says F positive, so it's fertility positive, to a negative cell. So it's just transferring material from one cell to another. But they're not mother and daughter. They can be cells, bacterial cells from the same species or even from different species. Conjugation can also happen with HFR cells, which is what is HFR? High frequency of recombination. When that happens, what means is that your F factor, instead of being in a plasmid, it is, and this is just a regular that I just showed you. So there is a plasmid, which is an extra chromosomal piece of DNA that doesn't incorporate into the chromosome and then duplicates and transfers to the negative cell, period. Okay, so then both cells have that plasmid. If you have an HFR, what happens? First, that plasmid gets incorporated into this bacterial genome, into in the F positive cell. So now, where is it? Here. That plasmid got incorporated. So you see how the plasmid now is here, incorporated into the chromosome of that bacterium. And now that bacterium can duplicate the genome and transfer part of this genome to the other cell. Okay? So part of it gets transferred to the recombinant negative cell. What's the difference between this and what I showed you before? That here, what is being transferred is chromosomal DNA, not plasmidial DNA. That's the only difference. And usually this happens with a higher frequency. Okay? What type of genes can be transferred through a plasmid or through a piece of chromosome? Different types, but usually the ones that have a lot of effect, that you can see the effect, are toxins that can come this way or can come in different ways to the negative cell. So genes that code for toxins that are proteins. And also for genes that code for proteins that are related to antibiotic resistance. Okay? All right. I'm saying okay because I imagine you understood all this. And if you didn't, watch the video again, watch a different video, come to my office, so you have many options, okay? The only option you don't have is to give up. Okay, this is just a genetic map showing this is a circular chromosome of an E. coli, and these are the genes that can be present in specific bacteria. These are like sizes, right? You have 3,480 kilo base pairs. So it's 3,480 times 1,000 base pairs. So just so you see how many base pairs, which are different, all these different nucleotides, right, in pairs that can be present in the genome of this bacteria. Transduction. What is transduction? Is another way to transfer horizontally DNA from one bacterium to the other. It can be generalized, a random piece of DNA, or it can be specialized when there is a specific bacterial gene that are packed inside, oh, we have something new here, inside a phage. So what's a phage? Phage is a virus. What is this virus doing? This virus is going to inject its pink DNA into a donor cell, into the donor cell. This is not a recipient cell yet. These pieces of viral DNA are going to incorporate into the bacterium. But when the phage is formed, it's still, you see how the purple is the bacterial DNA? It's still some pieces of bacterial DNA in some of their virions that are being produced. And then this phage injects in a recipient cell a piece of DNA that came from the donor cell, not from the virus. So the virus is just like the mail guy, right? He's just communicating, bringing a piece of DNA from one bacterium to the other. So when this genetic transfer happens through a bacteriophage, which is the type of virus that only infects bacteria, you call it transduction. And I told you what plasmids are, right? Self-replicating circular pieces of DNA, usually 1 to 5% the size of the bacterial chromosome, and often code for proteins that enhance the pathogenicity of the bacterium. Okay, we're done with this chapter. And I'll see you for chapter 10. Remember, chapter 9 is not included in our program. Have fun. See you later.